Hey guys, I apologize for being a little bit behind on lectures. I'm actually only one lecture behind because last week we only had two. But I had tons of problems with Zoom last week, including having this lecture that I'm about to do again right now completely deleted. I'd gotten all the way through the lecture. I was literally on the last page and Zoom shut down and I lost it. So I only kind of decided to call it quits. And now I'm going to go ahead and finish that lecture. And I'm apparently in here with a fly. I have to tell you this, even though you're not here to see me, but I used to teach most of the time in the biology building, urban and science building in room, was it 113? So it's the big theater type room. And there were always little fruit flies, like always. And was it 2 semesters ago? It's probably last fall. I was standing up there teaching and I could see this little fly flying around. And at some point, it flew by my mouth at the same time that I inhaled. I seriously inhaled that fly and it like hit the back of my throat, like I felt it. And I started like choking in the front of class. And everyone was just staring at me, like, what the, Then of course I had to explain, not choking to death, but I did just inhale a fly. I have a little bit of PTSD. Now when I see those little flying around flies, now I'm like, gonna inhale it. Anyway, there you go. There's your story for the day. All right. So we're gonna go ahead with nervous system 1, and then that will be from last week, and then I'll go ahead and finish up with the lectures that are supposed to be this week. Those will be released on time. Any other, I don't think I have any other announcements. If I do, I will email them. All right, let me share my screen. Let's go ahead and get this going. Hopefully it will not crash on me this time. I just don't think I have the patience to do it again. True story. Stuff around. What colors should we do? They're all so ugly. They're just yucky colors. All right, we'll try this one. Okie dokie. So fill in the blanks. This first one is going to be, this is the central nervous system, or CNS. It's not a terrible color. Okay, so the central nervous system consists of the brain and the spinal cord. And then we have the peripheral nervous system. Abbreviated PNS. And those are all of the nerves that are outside of the brain and the spinal cord. So I always kind of think about their relationship. It's like you've got the peripheral nervous system, which is in things like your fingertips. The peripheral nervous system is going to send information to the central nervous system, to your brain. Your brain's the processing center. It's going to process it and then send its decision back out to the peripheral nervous system for the muscles to act on the decision. Okay, so it's the whole fingers touch a hot stove. Message is sent from the peripheral nervous system to the brain, which says, holy crap, move your hand. That message gets sent back to the peripheral nervous system. The muscles receive the instructions to move. Okay. And there's a typo here. This shouldn't be CNS, not SNS. Okay. All right, so the nervous system receives sensory input. Sensory receptors in the skin, okay, out here, and other organs generate nerve signals that travel by way of the peripheral nervous system, where they're located, to the central nervous system, where they'll be processed. The central nervous system performs information processing and integration, which is what we talked about. It reviews and stores the information, creates appropriate motor responses. So it says, like I said, hey, that's hot. We need to move. Okay. At that point, the CNS, the central nervous system, generates motor output, tells the muscles to move. And those nervous system, or those nerve signals travel through the CNS through the PNS to the muscles, glands, and organs. So these muscles, glands, and organs, and anything else that has the ability to act on a nerve signal are called effectors. Okay, so these are the things that have the ability to actually act on what the central nervous system says to do. All right, 3 types of neurons. This first one, these are sensory neurons. They take nerve signals from the sensory receptor to the central nervous system. Okay, so you're going to find sensory receptors in all these, what do I want to say, all the rest of the body. That's not going to be part of the central nervous system. So organs have it as well. I mean, if you've ever had a kidney stone, or if you've had gallstones, or if you've ever had an internal injury, or anything like that, like you've had the flu and you've had stomach cramps, our organs also have these neurons that send and receive muscle messages or smooth muscle messages. Okay, and so then we have something called interneurons. Interneurons are only found in the central nervous system. So if you see this term, they're not out in the peripheral nervous system, only in the central nervous system. They sum up the information that's received from these sensory neurons, and they are going to communicate with the last group, which are called motor neurons. And again, these motor neurons are the ones that are going to cause a response. Okay. So we have interneurons that are going to communicate with these motor neurons. So the interneurons are like, okay, this is the decision that's made. This is how we're going to respond to this hot stove that we're touching. The motor neurons are like, okay, got it. I'm going to go communicate with the muscles to say to move their hand away. Okay, let's do these real quick. Axon terminal, this is the end where signals relayed. The cell body contains the nucleus and other organelles. The dendrites receive signals, and the axon conducts signals away either to the next neuron or to an effector. Okay. All right. Okay. Back in place. So real quick, I'm going to show you, actually, let me go back to this other page. Yeah, okay, this one's a little bigger. So I just wanted to point out here, which you have probably seen on the other page, but I just want to point it out, that we've got all 3 types of neurons illustrated here. You have the sensory neuron, the interneuron, and then you have the motor neuron. Okay, so these kind of represent the structure of all 3. You can notice that they are different. When it comes to the interneurons, the axon is not as pronounced as it is in your motor neuron, which looks like a scorpion tail. And then down here at the end, this is where you have that axon terminal, okay? And a lot of the things that are going to be passed as signals originate, I don't know if I want to say originate, they don't originate here, but they end up here, it's the terminal. And so like neurotransmitters in the brain, things like that are released here at the axon terminal into the gap that's between the axon of one and the dendrites of another nerve cell, okay? And we actually won't get into that in a ton in this lecture. Okay. All right, so let's talk about axons a little bit. As you know, this is where the information is passed on. So a neuron gets the message the axon's going to pass it on to the next. And axons can be either myelinated or non-myelinated, okay? So they might be protected by a myelin sheath, which are these light blue areas here, or they might not, okay? Which is what you see on the axon of this interneuron. So there's another type of cell in the nervous system called a glial cell. And those are the ones that produce the myelin, but we're not going to talk about them past me telling you their name. Write this down. We'll talk a little bit about how myelin affects signal conduction towards the end of this lecture. So kind of just here's some info on it and hang on to it. So when we talk about the central nervous system, you often hear about gray matter and white matter. Well, gray matter in the central nervous system is not myelinated. Whereas white matter is. Okay, so if you look at this diagram here where I have all these myelinated sections, notice that in between them, I have these non-myelinated sections. These little non-myelinated sections are called the nodes of Ranvier. And again, these are just the exposed areas of the axon itself. So they're non-myelinated areas, okay? All right, so nervous impulses are electrochemical signals that convey information. Electrochemical, so they're chemical, right? Because chemistry is the foundation of all of these things. The electro part comes from ions, okay? Which are extremely important in the body for a whole number of reasons, but some of the big ones are here. Sorry, still fighting a scratchy voice. So let's talk about some of those ions. Before I do though, I'm gonna give you some terms that we're gonna go over, but I'll tell you about them a little bit now. When it comes to a nerve impulse being sent and received, we have these steps. We have a stage called resting potential where nothing's happening. There's not any signal being sent or received. We go from resting potential into depolarization, okay? And then we go into repolarization, and then from there, we're gonna have kind of a short rest where the nerve resets to resting potential again. So I'm gonna go through those steps. And we're gonna start again with just what the baseline is, what's happening at resting potential when nothing's going on, okay? At resting potential, there's a charge of a negative 70 millivolts, okay? We're gonna watch that change during the depolarization part, and then it will reset at the end. But when nothing's happening, it does have this negative charge. Why does it contain a negative charge? Especially because if you look right here, the ions that we're talking about that are charged are positive. They have a positive charge. But the thing is, within the axon, there's also a bunch of negatively charged proteins. And so those other components that are inside the axon, such as those proteins, are what gives it an overall negative charge, okay? So we have sodium and potassium are going to be the ions that are playing this huge role in sending a nerve signal. The sodium is outside of the axon membrane. This is important that you remember what goes where I was writing. Okay. When the nerve cell is at resting potential, then this gate right here that lets sodium move back and forth from inside the membrane to outside is closed. Okay. We have potassium. The potassium ion is inside the axon membrane. And again, at rest, the gates are closed. Okay. All right. So if these lines here, these lines are like, the membrane of the axon, you see the sodium sitting outside with its gate, and you see the potassium inside with its gate. Okay. Let's move on. Maybe. So before we move on to this next stage about depolarization, we need to talk about action potential. So we were at resting potential, which is what we were talking about, where there's no signals being sent. Well, what happens when a signal is going to be sent? Okay. So the thing about action potential is that it's all or nothing. So the action potential is the process of conducting a nerve impulse along the length of an axon. Well, you can't have a little bit of a nervous impulse, okay, in the nervous system. You either have one that's strong enough to make depolarization happen, or you don't. And if it falls short, nothing happens at all. It's not like the gates open a little bit, okay? It's either big enough to cause depolarization or it gets ignored, essentially, okay? All right, so let's say that the cell receives action and action potential that is strong enough to cause depolarization. We're gonna have a change in charge, that we need to talk about now, okay? So at depolarization, we're gonna start with the sodium. The sodium, those gates, this gated channel is gonna open. Okay, so the sodium, this gate opens up here. At that point, we're gonna start dealing with a concentration gradient, and we're gonna be looking at diffusion, which is, if you remember, passive transport. So I'm gonna fill in some more sodium ions out here. Okay. It's crowded with sodium on the outside of the membrane. So as soon as these gates open, the sodium is super excited to go from where it's really crowded to where it's less crowded with sodium. It doesn't care that potassium's there. It only cares about getting away from other sodium ions, okay? So as soon as those gates open, the sodium diffuses from the outside of the membrane inside, okay? Using this passive process. Okay. From outside the axon to inside. Okay. So here's the thing. It's called depolarization. We're having a change in the charge, like I said. We've been at -70 millivolts when it comes to resting potential, right? Nothing's happening, it's at a -70. But now all of a sudden we have this flood of positive ions coming in. So now for a short time, we're gonna have both sodium and potassium inside the axon. That's enough to take our voltage from a -70 to a positive 35 millivolts. Okay, that's significant. However, this doesn't last terribly long. Okay. Slide actually rather than. Okay. Nope, did it happen? Hang on. Sorry, just having a minute. There we go. All right. So, like I said, this doesn't last very long because no sooner does the sodium get in here than it wants out again, or than the potassium wants out. Okay. So the sodium comes in through its channel, and then its gate for that channel closes off. So no more sodium can get in, and it's not going to get out. But now the potassium gated channel opens. Okay. I gotta move some stuff. All right. So now these are going to open up. We have the same thing with potassium. I'm just going to say it's going to move down its concentration gradient. So now it's going to go outside the axon. Okay. So we had our sodium that came in, its gates closed off for a minute during depolarization. We were at a positive 35 millivolts. But as soon as the potassium gate opens, the potassium is going to flow out following its concentration gradient. And that's going to repolarize or reset the voltage to a -70 again. Okay. All right. All right. So -70, positive 35, -70. Then we move into this little break I was telling you about, and it's called a refractory period. So this is a brief period when the part of the axon that was conducting a signal, it can't have any more signals sent to it. It can't respond to any actual potentials because it has to reset first. I'm squishing my words together, sorry. Okay. All right, so if you're wondering why we have to reset because we're back to a -70, you're right, we are. But our ions are totally screwed up. They're in the wrong place. The potassium is supposed to be inside and the sodium is supposed to be outside, but that's not what we have going on. So during the refractory period, we're going to reset our ions. Okay. The way that you can remember this is something called pumpkin. Pump K, a little plus N. So if you're trying to remember what goes where, you're going to pump the potassium symbol K in pumpkin. Okay. So to do this, though, if you notice, we've got the word pump going on here. That's because this is going to be an active process, meaning it requires energy. It might have been diffusion when it was moving along its concentration gradient, happy to not be in crowded conditions, but now we've got to force them to go back to where they belong, which means it's still going to be crowded, and they're not happy about it. So we've got to use our sodium-potassium pump to force the sodium through its channel, actually through our pump right here, through our pump. I'm going to force the sodium back outside. We're going to pump K in, pumpkin. We're going to send our potassium back inside. Okay, so that's what's happening with our sodium potassium pump. And once this is reset, then we can deal with another action potential. So we're going to reset the ions. Sodium goes out, and we know that we're going to pump K in. Okay. Once we've done that, we're back here at this bottom image where everything has been reset. We're back to the negative 70 millivolts and the ions are where they belong. Okay. And now we can start over in this process of depolarization and repolarization. If another action potential big enough to cause that depolarization happens. Okay, so obviously you're not going to be working in groups of two, talking about a drug that manages pain, how might it work, what part of the action potential might it affect. This is one of those things where if you are feeling like you're not 100% comfortable with everything that we've talked about, if you kind of give this some thought and work through it a little bit, it'll give you that practical application rather than just trying to memorize steps. Okay, but a couple of things that exist today, we have lidocaine, which is a relative of Novocaine. So, you know, you get in a dentist chair. Hang on, I spelled it wrong. C-A-I-I-N-E. Okay. Anyway, it tends to be topical, but it can be injected. And this is going to briefly, doesn't last, as you know. This is going to be something that briefly blocks the action potentials in a small area of the axon, okay? Or in a small area of the body. So the nerve cells, the axons of those nerve cells in a small area will be temporarily blocked from receiving action potentials. So you're numb because that action potential isn't being sent down the line, okay? Therefore, the nerves don't respond. So this is one of your local type anesthetics because it only affects the site, it's not the whole system. Then we have TTX, tetrodotoxin. Tetrodotoxin. Like I have to still sound things out phonetically. I think this is an O. Tetrodotoxin. Okay, so this is that pufferfish derivative. Neurotoxin. And it can be deadly because it works to paralyze all of the nervous system. It paralyzes the nervous system so that it can't send messages to the muscles so that the muscles can work. And when you think about that on the level of, like, you know, your respiratory system, that's a big deal because then all the muscles that are required in breathing are frozen, they're paralyzed, and so you can't breathe. And in large amounts, it can stop the heart and keep that from pumping, okay? So tetrodotoxin is potentially deadly. And if you are a Marvel fan and remember The Winter Soldier, as well as I do, the movie, I mean, I believe that tetrodotoxin is what Nick Fury used to make it look like he was dead, but he wasn't. again, because it causes the paralysis. All right, so question of the day, can eating pufferfish sushi kill you? Short answer is yes, if you have somebody that doesn't know what they're doing. There's only a part of the pufferfish that actually has this toxin, so as long as it has been removed prior to the sushi being made, then you're good. Yes, if, what do you call that, sushi chef? What do you call it? Yes, if, let's try that again. Okay, but as long as it's prepared correctly and they don't include any of the parts that actually have the tetrodotoxin, then you're good to go. All right, remember I told you that we were going to get back to the nodes of Ranvier and myelination and how it affects nervous signals? We're going to talk about that here. So there's two types of signal propagation, or two ways that signals can be passed along the length of an axon. The first is called saltatory, and this word is referring to kind of a jumping action, okay? What happens is basically what you see right here. The action potential is going to affect the nerve or affect the axon only in the non-myelinated areas. So that exchange of ions with our sodium and potassium, those are going to happen here. Okay, so you have this jumping type of signal propagation along the axon. It's not flowing straight through it like you'd think of water out of a hose, but rather kind of jumping from one point to the next. This actually allows the action potential to travel faster. Okay, and the ions exchange, if you remember what these little non-myelinated parts are called, at the nodes of Ranvier. Squish that there, okay? Then we have continuous conduction, and this is where the axon is non-myelinated. So there aren't any nodes of Ranvier because the whole axon is uncovered, okay? So this makes the action potential travel slower because instead of just being able to hop from one place to the next, it has to extend down the entire length. So ions are exchanged continuously along the entire length of the axon. And again, if you'll remember that in the central nervous system, we're going to have parts that are both myelinated and non-myelinated. Again, that white versus the gray matter. And when I talk about something moving faster or slower, we're still talking about fractions of seconds, so fast, so that even our continuous slower action potential, non-myelinated axons are still sending lightning fast. Okay, so last page. So we talked about our axon terminals. Come on, boys, you can do it. Axon terminals, and how we have neurotransmitters and other signals released from that part of the terminal into something that's called a synaptic cleft. Okay, so this gap exists between the two cells called the synaptic cleft. Okay, So the action potential stimulates the release of a neurotransmitter from the axon terminal into the synaptic cleft. Okay, and that chemical is going to relay the signal to the next neuron in line. Okay. Now, when the signal has reached its end, when it's gone as far as it's supposed to, or when the stimulus that's causing that signal to be made is gone, that neurotransmitter that's been released can only be stopped in two different ways, okay? The signal can only be stopped in two different ways, and that has to do with eliminating the presence of the neurotransmitter in the synaptic cleft, okay? So one of the ways that that can be eliminated is through an enzyme that breaks it down, or the axon terminal can actually reabsorb it. So let's say that you're getting a vaccination, right? As soon as the needle pierces your skin, you're going to feel it, and you're going to have action potential, and you're going to have nervous impulses that are relayed to the brain that are telling it it's happening. But as soon as that needle is removed from your arm, those signals stop because the source isn't there anymore. And so in order for that signal to stop being sent, the neurotransmitter that's been released into the synaptic cleft has to be broken down. Again, either through an enzyme present that breaks it down, or from the axon terminal of the sending axon being like, hey, we're good, we're done, I'm just gonna take that back, okay? There's a ton of different medications that function here. Some of the things that are like the serotonin type drugs actually keep the neurotransmitter in the synaptic cleft a little bit longer to prevent some of them from being taken up too quickly. There's a whole bunch of different ways that this happens because, again, you can have either too little neurotransmitter being released, you can have the neurotransmitter being picked up way too fast. There's other conditions where you have neurotransmitter released, but it's not very good at being picked up because there aren't enough receptors. So a lot of the drugs that deal with these types of things are actually looking at the synaptic cleft and how those signals are being received and picked up. Well, the same thing, sent and picked up, okay? I won't go into a whole lot of detail, but there are a lot of studies right now that are looking at depression, anxiety, bipolar disorder, schizophrenia to some extent. What else? A bunch of things that are related, showing that there are genetic markers that actually are responsible for a lot of that, for the reasons I said. Because you can have, your axon terminal that's just not sending enough neurotransmitter. Your body's not producing it, okay? Or you can release it and there are too many receptors on the dendrite that's going to pick it up with the next neuron, and so it gets picked up too quickly. Or it doesn't get picked up enough or not at all because you're missing receptors on that dendrite that should continue with that signal. And so because of that, again, there's a lot of medications that are being geared toward dealing with what happens at the synaptic cleft. You can be tested for it. My mom was. It's actually really interesting to find out. My mom has 3 different genetic mutations that prevent serotonin uptake. It severely inhibits serotonin uptake. It's fascinating to find out. I haven't had mine done, but knowing what her genetic makeup is on that it kind of can give me an idea of what I could be dealing with when it comes to that. 3 different serotonin uptake issues. Anyway, so it's really effective because when it comes to mental health, a lot of times practitioners are kind of, well, we're going to try this medication at this dose, and we're going to try this at that dose, and we're going to adjust them. And you can go through like a wicked adjustment period where you're just all over the place. The fact that they can look for these markers really helps to significantly narrow down the type of medication that you're going to respond to. It's amazing. So anyway, that all has to do with nerves and pulse serotonin uptake, not just serotonin, but in this case, what I was talking about. Yeah, okay, there's the end of this lecture, and I will have another with you for the other half of this nervous system here shortly. Thanks.